Second, I recently moved from University of Minnesota to South Dakota State University. So in this presentation, I will discuss our recent work on turkey hepatitis reovirus. So turkey hepatitis reovirus are member of family Reoviridae with a genome of 10 segments divided into large, medium, and small class. But I would like to focus, please pay attention on this outer capsid protein like sigma C cell attachment protein, sigma B S3 segment, and mu B M2 segment outer capsid protein that I will discuss in this presentation. So turkey reoviruses, we are working on these reoviruses from 2010, and before 2010, we have seen the enteric reovirus associated with the enteritis or enteric disease syndrome, and isolates that we have from such cases, mainly from the intestine with the diarrhea, we tentatively named them as turkey enteric reovirus. Later on, we have seen the cases of arthritis with the rupture of gastrocnemius tendons or digital flexor tendons in chronic cases, and the reovirus isolated from tendons from lameness cases we tentatively named as turkey arthritis reovirus. And most recently, we have seen the cases of hepatitis, enlarged liver with necrotic foci, and reovirus isolated from liver samples we named as turkey hepatitis reovirus. So I would like to emphasize here that this is tentative nomenclature just to differentiate these three different disease conditions or three types of reoviruses. So just to summarize here, we have enteric type reovirus, not a big concern for the industry. Then arthritis type reovirus, mainly infecting the older age turkeys, cause significant economic impact due to high culling rate. And hepatitis reovirus, mainly infecting the young turkeys with the moderate to high mortality also. So with the funding support from USPOULTRY and Egg and hedge funding, we did whole genome sequencing of 80 turkey hepatitis reoviruses and compared them with our reovirus database, which has mainly the more than 300 turkey arthritis reovirus sequences and nearly 20 turkey enteric type reoviruses, with the representation from all turkey producing states in the US and Canada. So we did phylogeographic analysis just to see any region-based grouping of these hepatitis reoviruses, and we divided all the states in 6 regions. So I would like to focus only few segments here. So first is the turkey M2 segment, the outer capsid protein. So if you see here, the horizontal is the time to most common ancestor, and the vertical part is just focus on these groupings, mainly on the nodes. So if you see here, like at each particular node, there is no one particular type of color. So that means we did not see any geographical clustering. So that means we cannot say that these are the grouping or isolates from Minnesota or these are from North Carolina. All are mixed together. And then second, we did not see any unique clustering of hepatitis reovirus. So they are mixed with the arthritis type reoviruses. And the third important point here is that this unique group also represents that they may be unique serotypes. So here we can say that there are 4 to 5 unique serotypes based on this M2 segment. And the fourth is this time to most common ancestor is around 1908. That means this segment have multiple introduction over the years. Same for S3 segment, no specific phylogeographic clustering and no grouping of the hepatitis reovirus only. So all are mixed together, and time to most common ancestor is 1980. The S3 segment, which is very important because of the cell attachment receptors. Same, we did not find any geographical clustering or the clustering of the hepatitis reovirus different from arthritis. But the important point here is that the time to most common ancestor is 2004. And that was the time where the new variants of chicken arthritis reovirus were emerging all over the world. So we hypothesized that there may be some unique changes in the S1 segment or sigma C protein of turkey enteric type reoviruses that change its pathogenicity and to cause it arthritis type or later on hepatitis type reoviruses. For rest of the segment, pattern was similar to S3 segment. So our conclusion here is, so then we did the serotyping of representative isolates in collaboration with AVSER, and based on this serotyping, there were 3 main serogroups of these 27 isolates, and 4 isolates were not serogrouped. So that also correlate with the sequencing information where we have predicted 4 to 5 unique serotypes. Then we did the pathogenesis study. At the time, we have seen the cases of turkey hepatitis. We were planning doing the comparative pathogenesis study of turkey arthritis reovirus in comparison with the two enteric type reoviruses that we are thinking, because the isolates from 2007 and 2008. So we were thinking that they may be true type of enteric reovirus. So we took that opportunity and included the 2 hepatitis reovirus also in that study to prove the cost postulate. So this is our experimental model where we inoculate the turkey poults at 7 days of age, 0.3 ml orally to follow the natural route of infection, and then kept them up to 35 days of age. We collected different tissue samples for real-time PCR and histopath. So all viruses replicate at 5 to 7 days in the intestine. So this is the vertical is the virus gene copy numbers and horizontal is days post inoculation. But this pattern was different in the liver. So only the hepatitis reovirus have high gene copy numbers, and some arthritis reovirus also showing the gene copy number here. In tendons, to our surprise, in addition to this is our Tar O'Neill reference strains, we have seen very high copy numbers of hepatitis reoviruses in tendons. And the same pattern was observed based on histological lesion scores in tendons, where we have seen the very high lesion scores are equal to the Tar O'Neill or reference strains in the both hepatitis reoviruses. Another interesting point from this study was this enteric type 2 from 2008, that also showing no lesions in initial stage, but at 21 days and 28 days post-infection, we have seen the tenosynovitis. So that means the arthritis type reovirus may be circulating before 2010. And also this may correlate with our S1 segment. Time to most common ancestor is like 2004. So maybe we have this type of arthritis reoviruses, or that changes started occurring at that point, but we were not able to notice those cases until 2010 or 11. So we re-isolated the virus and proved the cost postulate. And we published this paper recently in Avian Pathology. Then we planned the second study on comparative pathogenesis of turkey hepatitis reoviruses, where we have 10 isolates from different state and different age groups. We follow the same experimental model that I have mentioned previously, 0.3 ml virus was inoculated orally at 1 week of age. The only difference here is due to limited funds, we kept these turkey poults for up to 3 weeks of age. So we euthanize half of the poults at 2 weeks of age and the remaining at 3 weeks of age. So I will just directly go to the point on the histological lesion scores, where we have seen the same pattern as in the preliminary study, that we have seen very high lesion scores in tendons at 7 and 14 days post-infection. So in summary, based on the whole genome sequence analysis, turkey hepatitis reoviruses were closely related or grouped with turkey arthritis reovirus. There was no specific grouping of hepatitis reovirus, no geographical specific grouping, and both on the cross neutralization assay and sequence-based typing, we have seen the 3 to 5 main serotypes of these hepatitis reoviruses. And turkey hepatitis reovirus consistently induced tenosynovitis in the poults, and that lesion score is almost identical to turkey arthritis reoviruses. So the take-home message from this presentation is that based on all 3 types of studies, sequencing, serotyping, pathogenesis, it indicates that turkey hepatitis reoviruses are variant of turkey arthritis reovirus with increased pathogenicity. So that means this virus is now able to cause hepatitis at young age and mortality, as well as arthritis or clinical lameness in older age turkeys. We are continuously working on doing the whole genome sequencing to predict any new variants that may cause future outbreaks and may be different from the autogenous vaccines. We're also working on using this sequence information to determine the serotype and pathotypes just based on the sequence analysis only. And we are looking for funding support to conduct a long-term pathogenesis of turkey hepatitis reoviruses. We are also using the advanced machine learning techniques to find the markers that we can say that, okay, this is the enteric type reovirus and not a big concern, or this is hepatitis or arthritis type reovirus, and we should implement the control measures accordingly. And we are also working on live attenuated vaccine, another project funded by US Poultry and Egg. So we are very thankful to the US Poultry and Egg for their continuous support. With that, I would like to thank all my collaborators, and special thanks to US Poultry and Egg for funding this project.